Happy Wednesday, everyone. We are at the top of the hour, so we'll go ahead and get started. I'm Cheryl Rogers, the Director of Marketing here at Golden Helix, and as we're nearly ready to release Varseq publicly, we thought it very fitting to give you a glimpse into Varseq's user experience. Bryce Christensen, our Statistical Geneticist and Director of Service, will give you his perspective from the analyst side. Bryce?
Thank you, Cheryl. And thank you to our audience for joining us here this morning. We're really excited to announce the public availability of Varseq today. As we go along, if you have any questions or comments during the presentation, feel free to enter them in the question and answer panel in the GoToWebinar software. We'll do our best to answer as many of those as we can at the end of our presentation this morning. As we go along, we'll follow the general outline that you see here on the screen right now. We'll start out by reviewing a little bit of the background of Varseq. From there, we'll move on and look at a pair of interactive demonstrations within the Varseq software. To begin with, we'll take a look at analysis of gene panel data. We'll actually look at some cancer gene panels. From there, we'll move on and look at how you would approach family trio exome sequencing analysis within Varseq. We'll take a step back then, move out of the software, and talk a little bit about Varseq today and Varseq in the future, what we hope to be able to do with the software as time goes on. Now, we've publicly unveiled Varseq a little over a month ago on October 1st in a presentation given by our CEO, Andreas Scherer, together with Gabe Rudy, our Vice President of Product Development. That recording is available on our website at goldenhelix.com, and you can feel free to go download that if you'd like to learn more about the product. And just a few highlights from that presentation to point out. Our company was founded over 16 years ago now, and during that span, we've been very active in developing software in the genomics space. Our flagship product, SNP and Variation Suite, is widely used and widely cited. More recently, we launched our Genome Browse visualization product that has been downloaded and adopted by thousands of users around the world. And work on Varseq, I suppose if you look back in time, we really revealed Varseq in the middle of 2013 in an article published on Genome Web in which we announced our intention to create a new product that's really focused more toward the clinical market and the clinical analysis side of genome sequencing. And that leads us to today and to the public release of the all-new Varseq software. Now, I say all-new, but really it's built on very mature technology. We've taken the best pieces of our existing software and incorporated them into this new product together with some very specific optimizations that make it one of the fastest and most user-friendly as well as most powerful platforms available for analyzing DNA sequence data. Now, along the way, we've had a lot of help from a lot of great friends and great stakeholders. Here, I'd just like to point out a few of them who have given us feedback on the product and feedback on the user interface and on the user experience of a Varseq. And of course, I can't possibly list all of the names of everybody who has helped out with this, but I'd like to thank them all anyway. So let's talk about our first demonstration that we'll be seeing. Cancer panels and gene panels in general, I suppose. Gene panels are designed to target particular sets of genes that are known to be important for particular diseases. In this case, we'll be looking at a cancer panel. And so cancer panels are targeting frequently mutated regions of genes that are known to be important in human cancers. And there are a number of different providers who market various gene panels. The one we'll be looking at today is the Ion Torrent Comprehensive Cancer Panel. Now, as you analyze this type of data, you should understand and know that variants in certain genes may be useful to identify tumor subtypes, which can aid in patient diagnosis. There also may be some variants that are actionable or informative for patient treatment decisions. And typically, you'll find that analysis workflows for these sorts of gene panels tend to be somewhat repetitive. And that's one of the great things about Varseq, is that it's designed to enable repetitive workflows and to take out some of the human error that might be associated with having more complicated workflows that involve lots of clicking, lots of decision making. What you're going to see in Varseq today is that we can take a particular workflow, and after it's been done once, we can do it over and over and over again with additional samples in the future. So with the workflow we'll see today, there are three major things that it's designed to do. First, we're going to confirm that we have thorough coverage of all of the targeted regions that are covered by this particular panel, or all of the hotspots, as you might say. So we have a list of known hotspots that we want to make sure we have coverage of. Then as a second step, we'll evaluate any variants that are actually present at those targeted sites, those hotspots. And we'll review the sequence qualities on those and perhaps make a report. Then as a third major step, we'll evaluate any observed mutations that aren't necessarily in that list of hotspot regions, but may still be present within the sequenced area. And again, we'll review those and make a report on them. And when we're done, if necessary, we can update our analysis template. That's what will allow us to apply the same workflow to another set of samples in the future. And I'll show you how to apply that template then onto another set of samples. So with that, I think we're ready to go ahead and back out PowerPoint land for a moment and come over to Varseq. So you see now Varseq running on my desktop. And Varseq is designed around the concept of projects. And here, when you first launch Varseq, you see you're prompted either to create a brand new project or open an existing project. And I have a list of recently accessed projects here. And we'll start by looking at one of those. Give this just a moment to load. And we're now inside the Varseq environment. And before we move on, let's take a moment to kind of explore the workspace and see what we're looking at here. Anyone who has used our Genome Browse product or used our SVS product with Genome Browse integration will immediately recognize that here to the top right we see a Genome Browse visualization window where at the moment we have a very detailed view of a BAM file which contains sequence read alignments around this PIK3CA gene. To the left of that, we have this region of the screen called the filter chain window. This is where a lot of the magic happens in Varseq. As you explore the filter chain, you'll see some numbers. Like here at the top, this number 4007 is the total number of input variants that came into this particular project. And down at the bottom, this number 55 is the total set of variants that are passing through all of the selection criteria that are then outlined in these three separate windows. So each of these vertical stacks of boxes here contains a workflow outline. And at the bottom, we see exactly how many variants passed through each one of these outlines. Now what you'll find is that as you click on any one of the numbers in this table, that this data table at the bottom updates to represent all of the captured variants. So when I clicked on the number 4007, I see a listing of all of the 4007 positions that are found within the current VCF file. Let's look at the table here for a moment. As we scan through the table, we see To begin with, that we have data captured from the VCF file, so we know chromosome and position together with what are the reference and alternate alleles there. Moving across, we see some sample-specific data. Right now we're just showing genotypes together with some common quality metrics like read depths and genotype quality scores and so on. Now, you may wonder, what about all of the other data contained within a given VCF file? It's there, but it's currently hidden. If we wanted to see it, though, it's not a problem to select one of those fields and it will immediately come up where we can see it again. So we're not removing or omitting any of the data from the VCF files. We're just showing a selection of fields right now. Now, as we move across, we see some various annotation sources that have been added. There's this transcript annotation that tells us what gene is it in, what type of variant is it, the HGVS notations that give us a less ambiguous description of the variant. Now, clicking on any one of these columns of data gives some summary information where, number one, it describes the contents of the column, but also as we move down we see that it gives some details about exactly what is found there. So we see what proportion of these are misces variants, or what proportion of them are synonymous or splice site mutations, and so on. As we move across, we see we've also brought in some annotations from other sources, such as Cosmic. The Cosmic database is fully integrated. We curate that on an ongoing basis to give you the latest updates, so they'll always be available here. And as we scan through, we've also brought in a few custom annotation sources here. For example, this column, this summary of CHIP2 hotspots, this is telling us what are some of the targeted regions that we're looking for on this particular array. And what you'll find with Varseq is that if you're able to if you have any custom annotation sources that you want to use as part of your analysis, that it's very easy to integrate them. And as long as they're in either a standard text file, tab or comma delimited flat text file, or perhaps in a VCF file or a bed file, or any of a number of other common formats from bioinformatics, we can bring them in and work with them here. So now that we've explored the workspace a little bit, let's talk about our actual workflow. If I had just opened this project for the first time and imported this sample, because of the way this template has been built, I can see immediately, based on this set of filters, there are 32 variants at targeted hotspots that fail to meet our minimum coverage criteria. When I click on that list of 32, and maybe we'll expand it up a little bit bigger here, I can see it looks like maybe the VHL gene is poorly covered. We're getting a lot of depths here of 1, so very minimal coverage. With panels, you might be looking for minimum coverage in the hundreds or even 1,000 or more. So perhaps we want to make a report on these 32 that are less than 100. And to make that report, I can simply clone this table into a new tab, unlock that, and now this is a permanent listing of those 32 low coverage variants. Moving back to the main table now, looking at the hotspots that actually have a variant present in this data. As I look through here, I can see there are 7 variants at known cancer mutation hotspots. I can see what the allele fraction is of those variants. So for example here, about 11% of the reads are carrying an alternate allele. Here I see about 27% are carrying an alternate allele. Now this particular variant is in the KRAS gene. This is exciting to see. KRAS, of course, is very important in cancers, and it's clinically actionable. Patients who carry KRAS mutations are candidates for anti-EGFR therapy. They're known to respond better to that therapy than alternatives. Now, as I clicked here, you may have noticed also that the view updated. So I can see in the alignment in the BAM file, more details of exactly what's going on there. As I click in the BAM file, I can see summary information, 1,400 total reads. About 24% of them are for this mutant base, the T base at that position. Now as I click in the table, again, you'll notice that it's updating to show me all of the details about that particular sample, or that particular variant, excuse me. And as we scroll down, we see in this area, this is a listing of all of the cosmic reports for this exact variant. And we can see literally there are thousands of them, primarily coming from the large intestine. This happens to be a colorectal cancer case that we're looking at, so this makes a lot of sense. Now, moving through, I can also see here in the table all of that information from Cosmic. And if I want to explore it in more detail, there are embedded hyperlinks here where, for example, I can click and get a link to the PubMed abstract reporting this particular variant that I selected. And I can dock that web browser inside of Varseq. And also, maybe I want to look at the Cosmic entry for that particular mutation. So again, by clicking that, it will update the web browser interface so I can see this particular KRAS gene, the identifier used in Cosmic. And as we scroll through, we'll find additional links that we can use to learn more about that exact variant. So if I want to create a report on that table, I can make a unique spreadsheet for that one as well. And come back now and look at the final set of 16. These are variants that are not necessarily the hotspots, but within the targeted genes. And we've set up a certain set of filters here, where we can see there are maybe 65 of them that are non-synonymous variants that are rare in germline tissue. Of those 65, 16 of them are known in Cosmic, so I can capture this at different levels. But if perhaps I want to keep that set of 16 as my final report, and I'm ready now to export this so that I can share it back with the genetic counselors or the clinicians or whoever it is that needs to see this, I can take these 3 tables that are fully annotated with all of the information about these variants, and with a few clicks I can save this out as an Excel spreadsheet that will capture each of these 3 tables as separate sheets within the Excel workbook, I guess I should say. So if I save this onto my desktop and just call it Cancer Panel, November 5th, export, and it should be on my desktop now, but I can open it directly from here. We see now that here's the data exactly how I left it. I have the low coverage variants in one tab. I have the hotspot variants here in another. I can see this KRAS gene or mutation that we had been interested in initially. It's all right there at my fingertips, as quick and easy as can be, really. So coming back into Varseq now, let's suppose that I like this particular workflow and it's something that I want to use again in the future on another set of samples. If that's the case, it's a simple process from the File menu to save this project as a template. If it's been saved as a template, here I can give it a name, and I'll base it on this previous Cancer Panel 1. So there's 3 parallel chains. I'll call it the November 5th version, and save this. Now, if I close out of that particular project and create a new project, you'll see that the first thing that you're prompted to do when creating a brand new project is to select a template. You can choose an empty project template where you could build up your own set of filters. You could select exactly the annotation sources that you want to apply. or you can choose a designed template, and this is the one that I just created. If I select that now, it will open Varseq in the background, but you'll see that it says that it's waiting for data, and it's prompting me in the foreground to select data to add. So if I come in here and find a few more samples, and I've got a set of them right here. I've got about 4 VCF files. I can click through the process now, confirm the sample identifiers that I want to use and their affection status, and click the Finish button. This will take just a moment now to update. And when it's done, you'll see that sample by sample, it's created the same set of tables that we wanted and giving similar output throughout. So for this de-identified sample 1, we see there are 106 variants that are on that low coverage list. There are 4 that are among the more novel variants and 9 that are occurring right at our hotspots. And we see that list here now. Of course, we imported 4 samples, not just 1. So where are the other 3? You can access them just by clicking the little Next Sample button. This takes us over to sample 2, which happens to be the exact sample that we looked at previously. We can move then to sample 3, sample 4, and for each one of them, see the exact information that we're looking for. Now, if I were an analyst tasked with processing these samples as part of a clinical workflow, probably what I would want to do is review each of these lists, perhaps take any notes as necessary, and I could proceed then to export the tables either as Excel format or text files or some other formats that are supported and pass them along to the lab director. So let's take a little break now and back out of Varseq and come to PowerPoint for a moment. So let's move on now and talk about exome sequencing and Varseq. So we just finished a demonstration based on gene panels where you kind of know what you're looking for. Every gene on the gene panel is there for a reason. With exome analysis, you don't necessarily know what you're looking for as you get started. You have a much larger search space to work with. As a result of that, the workflow that you pursue becomes very important. The set of assumptions that you make about how the causal variant might present itself are very, very important, of course. Now, what you see on the screen right now is a diagram of a workflow that was put together by one of our SVS users for analyzing exome sequence data in family trios. This is a workflow that when it was first implemented based on Microsoft Excel and using manual processing, it took several days, if not a week or more per sample to fully implement. Within our SVS product, the same workflow takes about 2 hours or less to complete. In Varseq, now, you can apply this workflow in about 5 minutes to each new family trio once the template has been created. And so what we'll look at now is an example of how this exact workflow has been implemented in Varseq and some of the optimizations that come from that. Let's go ahead and close out of the slide again and go back to Varseq. We'll go ahead and launch this project. So as we look at this, you'll notice this particular project is maybe a little bit more complicated than the gene panel workflow that we saw. And if we expand this out so we can see the whole thing, let's talk about what you're seeing and why it's important. So with the gene panel workflow, we saw just 3 parallel columns representing 3 different workflows. Here, the layout has a few more levels in it. The thing to be aware of is that whenever you see side-by-side boxes, it indicates that it's three parallel processes that ultimately are using OR logic to connect them. So we're looking at the union of three things. So what we have here is a set of QC functions that start from 50,000 variants. There are then separate processes for homozygous, heterozygous, and homozygous reference genotypes. And ultimately, there is a certain number of variants that pass through each one, making a union of 44,000 variants. Now, those 44,000 that passed QC get passed to the next step down vertically. So side by side indicates OR logic. Vertical stacking indicates AND logic, or intersection logic. So these 44,000 are all getting passed into the variant workflows section, where you see 4 different workflows have been coded here. One is to look at de novo mutations. One is to look at rare recessive variants, at compound heterozygous variants, and rare heterozygous variants that may be inherited in a dominant manner. So as we look at this a little closer now, one thing to be aware of is that Varseq is designed to be very flexible in how templates are built and applied, and I won't go through the details on all of these. But for example, with the de novo workflow, it's built up so you can specify exactly what you're looking for in each sample. So you can drill in and say, I'm looking for either heterozygous or homozygous variants in the child, or maybe you really just want the heterozygous, so you can turn off that option and specify then that the mother strictly needs to be called homozygous reference. The father similarly should be called homozygous reference. And ultimately, there are 16 such variants that after going through a few more levels of quality control, we find just 10 that fit our criteria for a de novo mutation. I see that list of 10 here now at the bottom. And as I scan through, I can view some of the selected annotations that were applied for this particular workflow. I can see that all of these are missense variants, and that is actually part of the filter chain that was applied. Moving on further, I can see that all of them are extremely rare in the 1000 Genomes Project, in many cases novel. They've never been reported before. Similarly, in the ESP database, these are all novel except one that only has just an extremely low observed frequency previously. In this next set of annotations, this is coming from the database of non-synonymous functional polymorphisms. Here we can see, based on various predictive algorithms like SIFT, Polyphen, Mutation Taster, Mutation Assessor, and others, exactly how many of those algorithms think that each one of these variants appears to be damaging. So it ranges from 1 out of 5 to 4 out of 5, and so on. We didn't apply any filters based on that, but we could if we wanted. And ultimately, out here toward the end, we see this ClinVar annotation. ClinVar, of course, is a database that catalogs all of the known variants that are associated with diseases. It's in the public domain, of course. And ClinVar is one of our primary sources that we're providing for identifying, again, known mutations and mutations of clinical significance. In this case, as I look at this list, clearly there's just one of them that is previously cataloged in ClinVar. Scrolling through, I can see, based on this disease name, it's associated with something called, I believe, Meyer syndrome. As I click on the row, I can get some more details on that here. But also, I can get links out to external resources again. So if I click on the OMIM link, which is one of the many sources cataloged within ClinVar, OMIM can tell us more about this particular syndrome. We can read down and see it's a rare disorder characterized by mental retardation, dysmorphic facial features, and so on. And given that this particular child was referred due to developmental delays, this might be a very interesting variant to go back and review and learn more about. Now, beyond that, if we want to review some of the other findings here, let's take a look. So the recessive workflow identifies 24 variants that have expected population frequencies less than 1%, but are homozygous in the child and heterozygous in each parent. So the probability of these ever being observed homozygous in one individual based on those population frequencies is quite rare. And we can scan through and learn more about these and exactly what genes they're in here. But I believe none of them are in ClinVar, so there's nothing here that's known to be clinically relevant. But in the functional prediction, we can also see that at least one of them is predicted to be damaging by all 5 methods, a couple that are showing damaging in 4 methods. So we might want to look at a few of those and include them in a report. Also, we have this compound heterozygosity filter here, where it's showing 179 variants being part of a compound heterozygous event, that is to say, 2 or more heterozygotes observed within one gene in the child, but within each parent, at least one of them is observed in each parent, so each parent is responsible for one. So it's essentially equivalent to a recessive event in the child. Now, there's 179 variants that are part of compound heterozygotes. We also see just 82 genes. And in this gene viewer, we can click through gene by gene and just see a subset of which genes are contributing, which variants are contributing at each gene. So if we wanted to look at HK3, for example, we can scroll through and see exactly what's there. And let's move over just a little bit. We can see that in 1000 Genomes Project, one of these is previously unknown, one is very rare and only ever observed in Africans, and so on. And finally, then, we have the rare dominant workflow that's picking out 60 variants. So the process here is similar to what we saw with the gene panels. If I want to create reports on any one of these workflows, I can take those variants, put them into a table, which then can be exported in an Excel format or into a text file or into a number of other formats for reporting purposes. And I would say, again, the most interesting finding we have here, without question, is that one de novo variant that's showing up in ClinVar previously associated with a disease that has similar symptoms. Now again, if I want to apply the same workflow onto another sample set, it's the same process that we saw previously. If I go to the File menu and choose to save this project as a template, and I'll just call it the Trio Analysis November 5 template, I can apply it then onto additional samples. So create a new project, select the Trio Analysis November 5, and proceed. Just like before, you see initially it's waiting for data until I actually add the data. So here now, I can go find a different family trio. It needs to know which one is the child, who the parents are. So I'll fill this all in, and once it knows that much information, it can proceed to run this all through the template. And you'll also notice that there are progress bars, give you an idea of how long it's going to take to run through this. It's probably going to take about 4 or 5 minutes to get this all done, but you can click here and get an idea of what all of the dependencies are that it's waiting for us to finish this out. But once that's wrapped up, we can come back and see exactly what the results look like. So let's let that run, and go ahead and close this down, and come back to our slides for a moment. So you may have noticed as we were going along all of those different screens within Varseq. There's the filter chain box and the genome browse window, the details pane, the data table, the web browser, also Evernote integration, which we didn't touch on yet today. But all of these different windows can be independently moved around. They can be layered in tabs. They can be resized. You saw a little bit of that as we went along. But I just want to emphasize that this allows workspace customization that simply isn't possible in browser-based applications, especially when you have multiple monitors available. It allows analysts to set up their screens to see the information that is most important to them. Just as an example of that, this is a picture of my own desktop. It's the exact computer that I'm sitting in front of as I deliver this presentation today. One configuration that I really like is to take my widescreen monitor, turn it into portrait orientation, which then allows me to see multiple BAM files. In this case, it's the exact same family trio that we were just looking at, and it's centered right now on one of those de novo mutations where we can see down here in the child that there's some presence of an alternate allele that's not observed in either of the parents. We can see other annotations in the same window, and it doesn't require all of the scrolling that you might have to do if you were working within a single screen. Now, the other nice thing here is that by moving Genome Browse out, it gives a little more real estate to show other features like the filtering workflows, as well as the data table and the details window that shows all of the details that happen when you click on individual variants. So the flexibility is really a great bonus with Varseq. Now, Varseq today in version 1 is a fully featured variant annotation and filtering tool. Hopefully you've been able to see that and get a sense of it as we've gone along today. The interface is designed to be simple and intuitive, and the software infrastructure has been designed to be as fast and responsive as possible. Given that Varseq is designed to do one thing, which is sequence variant analysis, and do it very well, we've been able to achieve a lot of optimizations in the software that make it so fast and responsive. The speed of thought is the catchphrase that we like to use here. Also, you've seen that Varseq enables repeatable processes. You can build these workflow templates and apply them repeatedly over and over and over again to additional samples. Right now, Varseq is optimized for working with gene panels and exomes. Other applications where you're generally reporting on either individual probands or individual families. There's also the ability to work more in an interactive mode to explore different study designs when you get into research applications, and the software is capable of scaling up to whole genome analysis as well. I'd like to reiterate that the Varseq software is built on a mature technology stack and uses some of the best technology from our long history of providing bioinformatics software. So finally, I'll point out that we've adopted a very simple licensing model. We don't want to penalize you for being successful. We don't require you to pay per usage or pay per click, as you might find with some other software tools. Our licensing is based on a simple annual license fee that once it is installed in your lab, you can have unlimited usage throughout the license period. Now, in the future, I'll just point out that Varseq will only continue to improve. We expect to have a very rapid and response release cycle, and we want to have your input. So we'd love to talk to you about any features that you'd like to see added into the software. A few things that I can tell you about right now that are coming soon will be the ability to save variants to an internal database for use in future filtering. The enhanced project and variant note-taking capability that's coming. I mentioned Evernote integration in passing, but even beyond Evernote, we'll be giving you the opportunity to take notes on observed variants and be able to see those notes again if and when you observe the same variant in the future. Variant filtering based on custom gene lists, sample level QC statistics, that is, we'll be doing more computations at a sample by sample level. The ability to be part of existing pipelines in server environments. That is to say, we'll probably be giving some backdoor access into Varseq to enable use in command line type environments. And we also have some very extensive algorithmic customizations planned. The future is really exciting, and we look forward to working with our customers to make it really the best product possible. So if you have any questions or need more information about Varseq, feel free to contact us. You can email us directly, give us a call, go on our website, and let us know what you think. If you'd like to have an evaluation copy of the software, that's easily arranged. Simply go to our website and make the request. Okay, so just looking through here, we do have a lot of interesting questions, and a lot of them are going to be similar in some ways. But first question, we'll just kind of start at the top and go through. Are there any specific programs to generate TSF files? Right now, TSF files can be generated in all of our software packages at Golden Helix. So we can generate them from Genome Browse, which is completely free, by the way. So that might be the easiest way if you want to work with the format, as well as SVS and Varseq. We can read just about any standard bioinformatics format and convert it to TSF, together with a lot of other text file formats can easily be converted. Question next, so saving the project as a template basically saves the filter settings and screen tabs. It's a little bit more than that. It also saves all of the selected annotation sources. We didn't dive into that too much, but of course you can choose from dozens of different annotation sources in our data library. It keeps track of which sources you selected. It keeps track of which columns you wanted to have in your active view. And yes, it does save the screen tabs, and it saves all of the filter settings as well. And it essentially allows you to recreate all of your workflow except for the human interpretation aspect, which is never going to be easy. VUS analysis, question mark, variance of uncertain significance. That is one of those aspects that requires human interpretation, of course. We do have the ability to add in a number of other annotations beyond the few that we saw in the demonstration here today. But right now, in terms of uncertain significance, we definitely support a handful of different functional predictions. I know in clinical analysis, functional predictions aren't always well liked because they are just that, they're a prediction, and there's some error involved there. Do you provide template workflows with Varseq? Yes. So when you initially install Varseq, it will come with some standard template workflows. We will also provide training and teach you how to build your templates, and also we'll probably be offering template building as a service as well. So if you would like us to sit down with you and build a rock solid custom template, we can help you out with that. Filtering for strand bias. So that's something that's going to require having access to some kind of strand bias statistics, probably through the VCF file. Right now we're not doing any computation on the BAM file on the alignments, but if you have some strand bias statistic in your VCF file, we can absolutely use it as a filter. How do you decide the impact of a mutation? So that's something I kind of glossed over. It is important to point out, so I'll swing back here and just show you this. If we look at the full set of, say, 34,000 variants here, and come over to where we have the output from that new variant classifier. You'll notice that you can summarize this and see how many variants fall into each of the various categories, and it covers everything from synonymous variants to 5' UTR premature start codons, lots of different things in there. It's a one-to-one mapping on the impact. So the impact is really a kind of a simple summary of this. So I think I mentioned earlier that missense always map to medium. Stop gain always maps to high impact. Intron always maps to low. So it's really a shortcut so that when we were setting up these filters, I can just have 3 levels to choose from as opposed to having, say, here more than 20 or so levels to choose from on that filter. But you could set it up, of course, any way you want. Can you use Varseq to look at variants shared by a proband and his brother or uncle rather than a trio analysis? Yes, you can do that. And I'll leave it at that. It is possible. It's just slightly different startup on the workflow, and it might be slightly more difficult to automate that as a repeatable workflow, but as a one-off exploratory workflow, no problem at all. Can Varseq be used to analyze mitochondrial genome for variants? I believe the answer to that is a resounding yes. Does Varseq work with both TCF and VCF formats? On import, we convert VCF into our native TSF format. When you're done, if you want to take this final list of variants and save it back to VCF, that is fully supported. Do you have a program that converts from VCF to TSF? We already had that question. You can do it inside of Genome Browse or SVS or Varseq, and Genome Browse, of course, is the free and easy one to get to. Can we see all the previously done internal cases in the Genome Browse in the window of Varseq? So that goes back, I believe, to what was on this earlier slide on coming features, the ability to save variants to an internal database. We expect that will be coming very soon. Your examples were all from human. I work with sheep. Officially, Varseq right now, we're supporting human data, although I will tell you unofficially there's no restrictions on using other genomes. It should work just fine. Of course, you usually won't have the same level of rich annotations available in a lot of other genomes. So just be aware of that, but yeah, we won't stop you from using it with sheep. Can the software upload independent VCF files together? That's a question that I might need to hold off on answering. It definitely allows you to upload multiple simultaneous VCF files that are all part of a particular workflow. But if you have 2 VCF files for the same sample, right now I don't believe that it will merge them. Can we compare different trio data in Varseq? Yeah, I didn't show that. The examples I showed here, we just had one trio at a time. You might remember that with the cancer panels, we had multiple probands and we could jump through them. I could have done an analysis where I had several trios imported simultaneously and arranged things a little bit differently to find what was similar from one group to the next. That type of thing is possible. Any specific modules for clinical mutation analysis? That's an interesting question. The entire software package is designed ultimately for use in a clinical setting. As time goes on, we will be adding more and more, shall we say, optimizations in that regard. But no, right now there are no specific modules that are the clinical versus, say, research. I think we'll go ahead and just take one more question here, maybe 2 more before we wrap up. Can I use Varseq for diagnosis, like checking for known disease-causing variants? This, of course, is different than finding a new variant. the answer there is yes. We kind of saw an example of that here on the one workflow where I compared to ClinVar. And we could even look at it here again right now, just starting from this full list of 34,000. If I just wanted to come over to the ClinVar clinical significance, this is one option for looking at known disease-causing variants. You might have other resources that you prefer over ClinVar, but ClinVar is at my fingertips, that's why I'm showing it. But yes, if you want to set up filters that just start from some universe of variants for a sample and look at different databases of known clinical variants, that is supported. Another question that's been in here a couple times, do you produce any kind of final analysis reports? That is in the roadmap. I'll just leave it at that. It's something that we plan to support, although right now we already have clients planning to use it in clinical settings. They have their own reporting structure, so that's been less of an issue, but it is something that we're planning to implement soon. So with that, I think we'll go ahead and wrap up. Thank you all for your attendance today. Thanks for all the great questions. And Cheryl, did you have any final comments?
I do, Bryce. First, a big thank you to you for your time. A couple of just quick announcements. As Bryce did mention, the webcast recording and slides will be up on our website at latest tomorrow. Next webcast coming up December 10th, and that will be presented by Brian Davis of the NIH. Invitations for that will go out mid-month. And as Bryce also mentioned, Varseq, if you're interested, get on our website. You can request more information there. Thank you so much again, Bryce, and everyone have a great day.